Cell membrane made clear. Every single living cell faces one fundamental requirement just to exist. It absolutely needs a boundary. This boundary has to separate the highly organized interior of the cell from the completely chaotic outside world. Think about a tent for a second. If there are only poles, the frame, with no canvas, it can't hold anything in place, right? It doesn't define a usable space. Well, a cell without a membrane is a lot like that tent frame. Without it, the cell would lose its shape, lose total control, and instantly stop functioning as a living unit. So, how exactly does a living cell separate itself from the world without completely starving? It's a fascinating paradox. Think about it. Complete isolation means no nutrients can get in, which directly leads to starvation. But on the flip side, being totally open to the environment means losing all control over internal chemical stability. The cell literally has to find a way to exist in perfect balance between those two extreme scenarios. For a really long time, the old way of thinking viewed the membrane as just a static barrier, kind of like a solid brick wall. But a much better way to think about it is as a controlled routing system. It's definitely not just a static skin. Instead, it's a highly active, selective smart gate that is constantly making strategic decisions about what gets to enter and what absolutely needs to leave. That brings us to the core membrane design. It is quite literally built for a paradox. The physical design of this membrane has to perfectly balance two completely opposing forces: the strength to actually keep the cell together and the flexibility to interact with its surroundings. Let's compare a steel box to a plastic bag. A steel box is incredibly strong, sure, but it's entirely rigid. It cannot adapt. A flexible plastic bag, however, bends, reshapes, and yet still securely holds everything inside. A cell's boundary has to be like that flexible bag. To survive the constant dynamic demands of life, it has to be able to bend, repair itself, and adapt on the fly. There are 3 irreducible truths about this amazing structure. 1. It forms a controlled boundary. 2. It assembles itself automatically. And 3. It acts as a selective filter. The absolute core of this is a double-layer design made of molecules. The outer parts of these molecules are naturally attracted to water, while the inner parts completely avoid it. Because of this really simple attraction and avoidance, these molecules automatically organize themselves into a continuous, super stable double layer. A crucial part of this design is self-organization. This is the process where a structure naturally forms based entirely on the behavior of its molecules. A perfect, universal analogy here is how soap bubbles naturally assemble themselves in the air. Biology at this microscopic level feels a lot less like deliberate construction with tools and much more like natural organization driven purely by chemistry. And that self-organized layer leads us to another really crucial property: selective permeability. This simply means the membrane allows certain substances to pass through while blocking others based on their size, their electrical charge, and their shape. Think of a highly secure building entrance. A secure building doesn't protect itself by locking absolutely everyone out. Instead, access depends entirely on identity and size. The cell membrane works exactly like that filtered entrance. To maintain this delicate balance, the membrane survives by being organized but fluid. It's constantly shifting in small ways, while still keeping its stable overall identity. A bustling bazaar is a fantastic comparison. People move continuously through the market, changing positions all the time, yet the market itself stays completely intact and functional. The molecules in the membrane do exactly this. They drift and reorganize locally, without the entire boundary ever collapsing. So let's look at crossing the border naturally and how the cell manages all this traffic. Crossing this cellular boundary requires a highly strategic system of traffic management. There is a clear, escalating complexity to how things move in and out, happening in 3 main ways: slipping through naturally, needing a specialized protein doorway, or requiring powered active transport. Let's start with the absolute simplest method. This is where small electrically neutral substances just slip right through the barrier entirely on their own. This natural slipping happens because molecules are always in continuous motion. They naturally spread from an area of high concentration to an area of low concentration. It's just like if a bottle of perfume is open in a room, the scent naturally spreads outward until it fills the entire space. The cell relies heavily on this natural physical tendency for the free movement of small materials, and it accomplishes this completely without spending a single drop of energy. But what happens when transport needs help? That brings us to specialized protein doorways. You see, natural diffusion just isn't enough to sustain all of life's complex processes. Larger or electrically charged molecules are totally incompatible with the membrane's inner, water-avoiding core. So, to solve this exact problem, the membrane uses protein helpers that act as specialized doorways. This makes the boundary incredibly precise. These transport proteins act exactly like highly secure entrances that only unlock and open for recognized materials. This is what makes the membrane's selectivity a reality. It doesn't just block incompatible things, it actually provides specific, controlled pathways for exactly what the cell needs.
Moving on to energy and active transport, and how the cell actually shapes its internal environment.
There are crucial moments when a cell absolutely must move substances forcefully against their natural tendency to spread out. Moving with the gradient is easier, it's entirely natural, and it requires zero energy. But moving against the gradient, that is much harder, highly controlled, and it absolutely requires cellular energy. It's exactly like the difference between letting water flow naturally downhill versus carrying heavy buckets of water uphill. Forcing movement against nature's flow demands a significant, deliberate investment of energy from the cell. Now, this energy use is definitely not a waste. It allows the cell to shape its own precise internal environment instead of just being passively shaped by the outside world. Spending this energy is crucial for critical survival tasks. For example, the precise management of salt and water within living body cells ensures that those cells do not distort, swell up, or fail entirely when the outside chemical environment inevitably fluctuates. Let's pull this all together and look at the living control system, translating chaos into survival. All these separate components we've discussed actually function seamlessly as one devoted system. The controlled boundary ensures separation without total isolation. The self-assembled layer forms naturally, just from basic molecular behavior. Selective permeability dictates that some substances cross easier than others. The flexible structure allows vital movement without things breaking apart. And finally, the stable internal space means the cell keeps the exact conditions it needs to function. It is a beautiful system where everything works perfectly together to regulate exchange. Which leads directly to this really profound thought. The membrane translates outside chemistry into inside survival. Think about the outside world. It is noisy, highly variable, and completely unpredictable. A cell's interior, however, requires absolute continuity and stability to survive. The membrane acts as the critical bridge between these two completely different realities, making constant life-or-death decisions about exactly what information and material are allowed to pass through. Ultimately, structure creates function. A flexible, self-assembling, and highly selective boundary is the exact tool needed to take the chemical chaos of the outside world and successfully translate it into a thriving, stable living system. Without this dynamic gate, there is no control, and without control, there is simply no life. Which leaves us with a fascinating question for the next time we observe any complex biological process. How is the elegant structure itself making that remarkable function possible?